Very good afternoon everyone. So today we are going to study one of the interesting topic in molecular basis of inheritance, that is DNA fingerprinting, which was given by Jeffrey. So let's imagine something. There was a crime scene. So we don't have any kind of evidence. We don't have any CC cam footage, or we don't have any eyewitness. We just have a droplet of blood and two suspects. Now the police will use a technique called DNA fingerprinting and will find out who the real criminal is. This is case 1. Let's keep it aside. Now there is one more case. So there is a girl child and there is a woman A and woman B. The woman A is claiming that this child belongs to her. In the same way, the woman B is claiming that this child belongs to her. Now the doctors will use DNA fingerprinting and will find out who the real mother is. So this is how DNA fingerprinting is useful. So let's see. So if we talk about a DNA, 99.9% of our DNA is similar. So between you and me, 99.9% of DNA is similar. You might ask me, sir, you are saying 99.9% of DNA between you and me is similar, but why don't we look alike? Because of the remaining 0.1%. Because of difference in 0.1%. This might look very small, but this is a very big thing. Because if we take human genome, we have around 3 billion base pairs. 3 billion base pairs. So in 3 billion, 0.1% means 3 million base pairs. So this is the reason why this is not a small number. This is a very, very big number. This is the reason why we are different. Because there is a difference of 3 million base pairs between me and you. That's the reason why we don't look at it. So now this variation is because of DNA polymorphism. 2000 years ago, let's imagine something. 2000 years ago, there is a person called A. His DNA got mutated and it is inherited into his children, and his children inherited into their children, and it passed by generation after generation. And after 2000 years ago, after 2000 years, a certain population will have that mutated DNA. Let's keep it aside. Remember this. There is a man whose DNA got mutated and it is passed generation after generation, and after 2000 years, a certain population has that mutated DNA. Now there is person 2 called B. Now he has also mutation, but at a different place. So in the same way, what happened to this person, we'll see in this person. So there, after 2000 years, there is a certain population with this DNA mutation. So we have two kinds of DNA here. One is DNA, one DNA which is mutated in a different place, and another DNA which is mutated at another space. So these different forms of DNA is known as DNA polymorphism. Different forms of DNA are known as DNA polymorphism. So if we take this DNA, here we have coding regions and non-coding regions. So we are talking about mutations. If mutation happens in these coding regions, it will be eliminated because it is harmful to them. So the organism or the species will be eliminated. So the mutations won't take place in the coding region of DNA. So the mutations always takes place in non-coding regions. Non-coding regions called satellite DNA. satellite DNA. So here we have repeated units. Repeated units called VNTRs. Variable number tandem repeats. So in a normal DNA, in a coding sequence, we have ATCG, CAAGAPG, something like this, which is continuous. But here in satellite DNA, what we can see is do you find any difference between both of them? Yes. Here it is continuous. There is no sequence at all. But here, if you see, same sequence is repeating again and again and again. So that is what variable number tandem repeats is. So this repeats, whatever we are saying, is different for different individuals. Now one person A might have this, and the person B might have just this. That's it. Some might have 3, some might have 4, some might have 2, like it depends. So if you carefully observe, here we have 2, here we have 3. So when there is 2 and 3, the length of the sequence is also different. The length, the fragment length differs. So this is the base of our DNA fingerprinting. So I will tell you how. So now we have a blood group, a blood draw in a crime scene. So we will find the DNA fingerprinting of it, and we will get some bands like and there is a suspect A and suspect B. Now he has a bands like this, and B has similar bands with the droplet. Now who is the real killer? A or B? Because why? Because this is matching with B. This is the reason why we can easily find out who the real culprit is. So if you see here, 99.9% of DNA between us is similar. There is only 0.1% is dissimilar. You might think that 0.1 is very small, but it's a very big number because human genome has around 3 billion base pairs in it. In that 0.1% means around 3 million base pairs. That's the reason why you are different and I am different. And this 0.1% is because of DNA polymorphism. DNA polymorphism means different kinds of DNA, different forms of DNA. So if we take DNA, there are two regions in it. Coding regions and non-coding regions. In coding regions, if mutation occurs, it will be eliminated because it is harmful for the individual. So the mutations won't occur in the coding regions. So the mutation occurs in the non-coding regions. It doesn't matter at all. When there is a mutation in the non-coding region, it doesn't matter. It is not that much harmful for an organism. And so the mutations occur in a region called satellite DNA, where we can see repeated units in it. So we call those repeated units as VNTRs, means variable number tandem repeats. Write down variable number tandem repeats. So if we take a normal coding sequence, we'll have a which is not a sequence form, like it will be in a normal way. But if we take this VNTRs, a single sequence is repeated again and again and again. So that repeated sequence is different in children. Some might have 3, some might have 2, some might have 6. So it depends. And the length of the fragments will also differ. So this is what about the DNA. Now let's see the real process behind the DNA fingerprinting. So the first step is sample collection. Sample collection. They might be anything. They might be blood drop, or they might be hair, nails, or saliva, or they might be semen, etc. They might be anything. Now we collected the samples. And the next step is DNA extraction. The next step is DNA extraction. So now we collected a blood drop. For suppose we collected a blood drop. So if we take a blood drop, what will be there in the cell? There are different kinds of parts in it. So we need to remove them to get the DNA from it. Only then we can move further. That's the reason why we are doing DNA extraction. So here there are many processes in DNA fingerprinting. We are not going to see how they are made. Like we don't see the process in it. We will get to know why we are doing this. So why we are doing DNA extraction means to remove the unnecessary part of it. We will get the purified DNA after DNA extraction. That's our main objective. To get purified DNA. First we collected the samples. And then we extracted the DNA from it. And the next step, digestion. So now we will digest the DNA product. Now we got the DNA product. So if we take 500 ml, 500 not ml, 2 ml of blood, we will get around 1 ml, not even 1 ml, 500 microliter of DNA from it. It's not purified DNA, still it has some other things. So now we got a DNA and we kept that in MCT. And to that MCT, a small tube, MCT means a small tube. To that MCT, we will add an enzyme called restriction digestion enzyme to it. And we will add that enzyme and we will incubate it at a certain temperature for few hours. So now why we are doing this? Because DNA is a very big and long thing. So we need only a few parts. We are studying about non-coding regions. We are not studying about coding regions. We are studying only on non-coding regions. So this restriction digestion enzymes acts like a scissors, and they will cut the unnecessary part and they will give us the only required part of it. That's the reason why we are doing digestion. By restriction enzymes. So we have collected the sample and we have extracted the DNA from it, and then we have digested the product. We have digested the DNA using restriction enzymes. So still the DNA which is digested with the help of restriction enzyme is in the small tube MCT. Now we will pipette it out. Now we will take a small amount of the digested product and we will cast them in a gel. So this process is known as gel electrophoresis. So now we have pipetted out the digestion product of DNA and restriction enzyme. Now we will cast them into a gel called agarose gel electrophoresis. So it is started with the help of electricity. So now what happens is the smaller fragments travels faster and the larger ones they travel slow. And one more thing, so DNA is a negatively charged particle, so it moves towards positively charged side. So now we got the DNA fragments. We suppose this as agarose gel. So this is the negative side and this is the positive side. So now here we have wells. And now we have casted the digested DNA product in it. And after 2 hours we will pass the electricity in this side and this side. We give the electricity to it, and after 2 hours or 1.5 hours, the DNA will travel and will get bands like this. So this can be observed under the UV light. So this gel is very delicate. So we cannot keep the materials there for longer time. So we need to shift it with a technique called southern blotting. So now we have casted. So now we have taken the digested product and we have casted it into these wells and we have passed the electricity. So now we will get bands. The smaller bands will travel fast and the larger bands will travel slow, and the DNA, as DNA is negatively charged, all the particles move towards the positively charged end. So this gel is a very delicate one, so it's like a very gelish type. So, and it is also harmful. It has PTBR in it. Now we will carefully take this gel and will transfer it into a nitrocellulose layer. Now we will carefully transfer this gel into a nitrocellulose layer, and this technique is known as southern blotting. So first we have collected the samples, which might be blood, hair, nails or saliva or semen. So first suppose next we have taken the blood. So now there are different kinds of parts in a cell. So we need to remove them. Only then we can get the DNA. So that's the reason why we are doing DNA extraction. After doing DNA extraction, there are many processes. Like we can have phenol chloroform method and iodine chloroform method, and those methods take around 24 to 48 hours. It's a time-taking process. But recently, the main scientists or the students who are working in laboratories, they use a kit, DNA extraction kit. With the help of DNA extraction kit, we can easily extract the DNA in 45 minutes. So it's a good thing. And now we got the purified DNA. And as we got the purified DNA is a very long thing. So we don't need the coding regions. We just need the non-coding regions. So we will cut them using a scissors called restriction enzyme. So in order to cut them, we will mix the restriction enzyme with the purified DNA. So we will incubate it for a few hours, and after that the product we will cast them in these wells of a gel and we will pass the electricity. Now we will get the fragments. So this gel is a very thin in nature. It's a very fragile in nature. So we will transfer this gel into a nitrocellulose layer. And this process is known as Southern blotting. Now, the next step is hybridization. Now we will add a probe to this. So now we have different kinds of fragments. So this restriction enzyme, like when it is cutting, it might also cut the non-useful part. So here we can see the non-useful parts and the useful parts too. So what we do is we will do the hybridization. We will add the radioactive elements to it with the help of a probe, a radioactive probe. Radioactive probe, which is complementary to the fragment. So now we will add the radioactive probe to it. After adding this, the next step, radiography. So now we cannot see the radioactive elements. So now we expose them to the X-rays. So now we expose them to the X-rays. So now we will get two kinds of bands, a dark band and light band. DNA is colorless. So the non-useful part is not needed. The useful part is only needed. The useful part is dark. The dark colored bands are because of radioactive probe. So this is the process, and now we got the bands. And now we will compare these bands with the suspected ones. Or we will compare. So if we suppose these bands are of a child. So if you remember in the beginning I told you that there is a child and two mothers. So let's suppose these bands are of a child. And we will compare these bands with the mothers, mother A and mother B. And we will see if these bands are almost closer to mother, then this child belongs to her. Or if these bands are closer to her and closer to B, this child belongs to B. This is how it is. This is what DNA fingerprinting is. And that's it. In the next video, we will talk about gel electrophoresis. This gel electrophoresis needs a separate video. So it cannot be explained in a single class. So in the next class, we will discuss about gel electrophoresis. Thank you.